Hello students, I hope you all are doing well and studying. In previous lecture, we have started with studying the types of asexual reproduction methods, and we mainly have studied about fission, budding, fragmentation and regeneration, and at the end we have studied about sporulation. Now, in today's lecture, we are going to study the remaining type of asexual reproduction, that is vegetative propagation. Vegetative propagation simply means development of a new plantlet from a vegetative part of mother plant. It is a method of multiplication in which a vegetative part grows into an independent plant under suitable environmental conditions. The vegetative part or organ of plant can be any of the root, shoot or leaves or any part of the plant body. This vegetative part, which is participating in vegetative reproduction, is also termed as propagule, or more particularly vegetative propagule. The propagule which is participating in asexual reproduction should have adequate reserve food material and at least one growing point at which it can give rise to a new plantlet. Methods of vegetative reproduction are broadly divided into two types. First, the natural methods, and the second, artificial methods. Artificial methods are also referred as horticulture methods. Now let us start first with the natural vegetative propagation methods. In these methods, some vegetative part of mother plant such as root, stem and leaves act as propagules. The propagule grows to form an independent plant under favorable conditions. The natural vegetative propagation can be studied under various headings based on the part of plant which is participating in the process. So the first type is tuberous root. In some plants like sweet potato and dahlia, the underground roots store plenty of reserve food and become swollen. Such swollen roots are called tuberous roots. The tuberous roots are modified adventitious roots for food storage and vegetative propagation. These roots in some plants have adventitious buds on their surface, and these buds sprout under favorable conditions to produce leafy shoots which are termed as slips, and ultimately they also give rise to adventitious roots. The sprout can be separated and planted for commercial cultivation. The tuberous roots may be produced singly or in clusters. The example includes sweet potato, dahlia, asparagus. Next, stem part or shoot part. Based on the location of stem part at which the propagule is forming, there are four types: underground stem, subaerial stem, aerial stem, and bulbils. The underground stem further divided into three parts or three types: tubers, rhizome, bulbs. The subaerial stem are also three types as runner, offset, and sucker. So let us discuss them one by one. So first type is underground stem. This stem is non-green, food storing and perinating structure present below the soil surface. Underground stem can be of tuber, rhizome, or bulb type. So the first type is tubers type. In plants like potato, the underground stem produces axillary or extra axillary branches. The tip of such branches becomes swollen due to the storage of reserve food, and ultimately it is called a stem tuber. A stem tuber has many notches on its surface called eyes. Each eye is actually a node and consists of one or more small axillary buds and reduced scale leaves. Under favorable conditions, one of the axillary bud forms new plantlet. So here the example is potato. The next type of underground stem is rhizome. So rhizome is main underground perennial stem that grows horizontally, and it is observed in several types of plants. The buds present on nodes of the rhizome take part for formation of new aerial shoot during favorable conditions. And once we separate this each part of the stem and we cut down into multiple pieces, that each piece can give rise to a new plantlet, provided that each piece is having this type of buds on it. So these buds ultimately giving rise to this type of shoot part. The example includes ginger and turmeric. The next type of underground stem is bulb. In plants such as onion and garlic, the bulb is the unit of vegetative propagation. It is a short underground stem reduced to flattened disc with compressed nodes and internodes. The modified fleshy scale leaves may bear axillary buds, which again can be of two types, the apical bud and lateral bud. The apical bud will produce leaves and a flower, while the lateral buds will produce new shoots. As the plant grows and develops, it will form a new bulb underground. So, the example of such plants includes onion and garlic. So, here you can observe this is the reduced stem part, this region, and onto this we can find this apical bud. This is apical bud, and these are the regions where we can observe the lateral buds are developing. So this apical bud, it is giving rise to flowers and leaves, while this two lateral buds, they are going to form the new shoots, and gradually as they will grow up they will get separated from this, and there will be more number of plantlets out of it. The another type of stem is subaerial stem. In some plants, the stem is partially above the ground and partially below the ground. In fact, it is a weak stem which takes support for creeping purpose or for spreading purpose. So, this type of subaerial stem again are of three types. The first type is runner. Runners are slender subaerial branches with long internode regions, and these runners arise from the lower axillary buds of the stem. These axillary buds, they are generally situated close to the ground, and their runner, which is a thin and long structure, creep horizontally onto the ground. The runners produces shoots from nodes, and these nodes are having mainly two axes. The upper axis will give rise to shoot part, while the lower axis will give rise to root part. So after getting detached from this parental body, this node region which has generated a shoot and root will gives rise to a new plantlet. So here you can observe initially this plant has developed this runner, then at this particular point it has given rise to the shoot and root. So later on this part will get separated. So the example includes lawn grass and mentha. The second type of subaerial stem is offset. The offset is found in aquatic plants and it is just similar to runner. The only difference between this offset and runner is that the offset is shorter and thicker than the runner. Offset produces clusters of leaves above and clusters of roots below its axis. Breaking of offset helps in propagation of plants, and this type of mechanism we generally observe in plants like water hyacinths. So example includes water hyacinth and water lettuce. Now in this, the water hyacinth is also referred as terror of Bengal. The reason behind this, the water hyacinth was introduced in Bengal because of its beautiful flower and shape of leaves. However, it turned out to be highly invasive aquatic weed which has spreaded throughout the water bodies. Now, this weed drains high amount of oxygen from water and that leads to death of fishes and other animals. As we know that fish is supplement food of people in Bengal, and due to this weed growing in water bodies, it has caused the death of fishes and that has led to food scarcity. And that's why this weed water hyacinth has been termed as terror of Bengal. The third type of subaerial stem is sucker. The sucker is an underground runner and it generally arises from axillary bud of underground part of stem. These suckers, which are also called as underground runners, gradually they grow upwards and produces new shoot, and that's how they lead to formation of new plantlet. So the example mainly includes chrysanthemum or garden chrysanthemum, pineapple, and banana. The next type is aerial stem. In plants like sugarcane, the nodes on segmented stem consist of several buds. So you can observe them here. These buds on each node can give rise to a new plantlet. In plants like cacti, the aerial part of stem develop into new plants only if that part of stem falls onto the ground, and that serves as mean of vegetative propagation. These are the two examples, the cacti and sugarcane we can consider for aerial stem giving rise to a new plantlet or aerial stem participating in vegetative propagation. The next type is bulbils. Bulbils are spherical multicellular structures produced in axil of foliage leaves. These large sized fleshy birds, they are specialized for vegetative reproduction in several plants. Bulbils grow to form new plant only when they shed and fall on ground. In plants like agave, bulbils are modified floral buds that developed from the floral axis. So example includes agave and lily. Next, leaves. In plants like bryophyllum and begonia, vegetative propagation takes place with the help of leaves. In Bryophyllum, leaf is succulent type and having notched margin. An adventitious foliar bud is present in each notch at the tip of lateral vein. These buds start sprouting on leaf to form leafy shoot and adventitious roots. When such sprout fall on wet soil, they develop into independent plant. So the example here includes Bryophyllum and Begonia. So all these were the types under natural vegetative propagation. Now we move on to the next part, that is artificial vegetative propagation, and as I said, it is also called as horticulture. So horticulture basically a discipline of agricultural science which is dealing with development and improvement of vegetable, fruit and ornamental plant varieties. The artificial methods are of two types. The traditional method, which are further divided into three types: the cutting, grafting and layering. The second type of artificial vegetative propagation, the modern technology. So micropropagation and plant tissue culture techniques are generally used to improve the crop varieties and to improve the yield or production of various crops. So these techniques, they are referred as modern artificial vegetative propagation techniques. Let us now discuss about them one by one. So first is cutting. In cutting process, a vegetative part of a plant is taken to form a new plant. That vegetative part can be of stem region, and if it is from stem region, it is termed as stem cutting. Or alternatively, the vegetative part can be from root region, and in that case, it will be called as root cutting. The second type is called grafting. In this method, a small branch of a plant is inserted into the stem of rooted plant. Both of this plant should be from the same species or allied species, and then only the grafting will be supported. As a result of insertion, fusion of tissues takes place and both these plantlets will grow as one plant. The inserted plant here is called scion or graft, and the rooted plant is called stalk. The third method, layering. A soft basal branch is defoliated in the middle and a small cut is given. The injured defoliated part is then covered under a light layer of moist soil to develop roots. This grounded branch is called as layer, and that's why the technique is termed as layering technique. Later on, as roots are developed, it is separated and planted. These are all the traditional methods practiced in horticulture for improvement of crop varieties or getting the desirable characteristics into crop varieties. Now next we will study about the modern technology used for artificial vegetative propagation, and it is termed as micropropagation. So micropropagation is one of the method adapted in plant tissue culture technology, and this technique is generally used to produce millions of clones of desired plant variety from a small piece of plant part. So this is an outline of micropropagation. Let us understand the steps involved in micropropagation technique. So micropropagation, it begins with selection and maintenance of stock plants for initiation of culture. So stock plant is any plant which is having the desirable characteristics. From this particular plant, we choose an explant. So explant is nothing but it can be any part of the plant, like it can be a shoot apical meristem, it can be a region of stem including certain nodes and internode regions, or it can be probably a leaf part, or it also can be from root part. So virtually the explant can be any plant part which is having living tissues. So after selection of this explant, it is excised from the mother plant body and then shifted into the laboratory. In laboratory, the explant is surface sterilized. So surface sterilization is mainly done in order to remove any possible pathogens present onto the surface of that explant. So for achieving this surface sterilization, different types of chemicals like alcohol or sodium hypochlorite or calcium hypochlorites are used. And once the surface sterilization is done, this explant is washed with sterile distilled water. Then it is shifted onto the nutrient medium. So nutrient medium here, this is nutrient medium. So nutrient medium mainly includes various types of macro and micro elements which are necessary for growth of the plant, and they are also supplemented with the hormones, phytohormones. So here in first step, the target is to produce multiple number of shoots, and that's how the concentration of hormone is maintained in such a way. So as the number of shoots keep increasing, these shoots are further excised and they are shifted into the other containers. Once desirable number of shoots are obtained, then the next step is called rooting step, where those produced roots are subjected to the nutrient medium containing phytohormones, giving rise to roots from the developed shoots. So once the rooting process is done, then the complete plantlet has produced, and now these plantlets are shifted for acclimatization. Acclimatization is nothing but it is a method, a step by step process where these plants which have been grown into laboratory conditions are now subjected to exposure of natural environmental conditions. So this process is also referred as acclimatization or hardening process. So here the newly produced plantlets then shifted into the greenhouses. So in greenhouses also the conditions are quite controlled, but not as optimum as in laboratory. So after few months of growth inside the greenhouse, these plantlets then shifted into the shed houses, and in shed house they again grow for several days or several weeks and they become mature. This is how the acclimatization is completed, and now the plantlet is ready for selling into the market. So this type of plantlets, they are being sellable. So that's it for micropropagation as well as the asexual reproduction type vegetative propagation. So at this stage we have finished with all the types under asexual reproduction. In next lecture we will start with the sexual reproduction. Till then, goodbye. Don't forget to send your comments or queries onto my Telegram channel. So the link to join that Telegram channel I have mentioned into the description. So please go through it. Goodbye.